This is the second part of the materials over Chapter 11. Continue talking about how to handle microbes. In this portion of the presentation, we are talking about drugs, the microbe-host interactions, and the elements of chemotherapy. This presentation relates to the use of antibiotics for control of microorganisms as outlined in Chapter 11 of your text. So please read and understand the materials in that chapter. I'm going to go through this presentation. I really have very little that I will add beyond the content that's on these slides. The objectives: explore the advantages and disadvantages of using antibiotics to control microbes in the body. Identify some of the important antibiotics used to treat diseases and indicate how these drugs achieve the antimicrobial activity. Discuss the problem of antibiotic resistance with reference to its origins and its implications. Additionally, there will be some material on the history of the development of antibiotics. You should know some of the key names such as Fleming and Domack. So, the origins of antimicrobial drugs. Antibiotics are common metabolic products of aerobic spore-forming bacteria and fungi. And what they do is they inhibit other microbes in the same habitat, so there is less competition for nutrients and space. Bacteria in the genera Streptomyces and Bacillus are involved, and by the same token, molds in the genera Penicillium and Cephalosporum are also involved, and these two compete against each other. Basically, bacteria make antifungal types of materials, and molds in general make antibacterial materials. This table shows some aspects of chemotherapeutic drugs, just some terminology, so make sure that you understand what a chemotherapeutic drug is, which is any chemical used in the treatment, relief, or prophylaxis of disease. Prophylaxis is the use of a drug to prevent infection of a person at risk. Antimicrobial chemotherapy is the use of chemotherapeutic drugs to control an already existing infection. Antimicrobics, just an all-inclusive term for antimicrobial drugs regardless of its origin. Antibiotics are substances produced by the natural metabolic processes of some microorganisms that can inhibit or destroy other microorganisms. Semi-synthetic drugs, drugs which are chemically modified in the laboratory after being isolated from natural sources. Synthetic drugs, the use of chemical reactions to synthesize antimicrobial compounds in the laboratory. So these are fully artificially produced antibiotics. Narrow spectrum or limited spectrum, that is a term used to apply to some antimicrobics that are effective against a limited array of microbial types. For example, a drug effective mainly on gram-positive bacteria as opposed to gram-negative. And then there are broad-spectrum or extended-spectrum drugs. And these are antimicrobics that are effective against a wide variety of microbial types. For example, a drug efficient against both gram-positive and gram-negative bacteria. Now, in selecting the ideal antimicrobial, what we have to do is look for selective toxic, we hope that it is selectively toxic to the microbe, but non-toxic to the host cells. We'd rather that it be microbicidal rather than microbistatic, relatively soluble and functions even when highly diluted in bodily fluids, remains potent for a long enough time, and is not broken down or excreted prematurely. That is, we don't want to have to constantly be dosing. Not subject to the development of antimicrobial resistance, complements or assists the activities of the host defenses, remains active in tissues and body fluids, readily delivered to the site of infection, not excessive in cost, and does not disrupt the host's health by causing allergies or predisposing the host to other infections. So those are just some of the factors to consider when selecting an antimicrobial. So let's talk about selectively toxic. Drugs should kill or inhibit microbial cells without simultaneously damaging the host cells. The issue that comes into play there is that as the infectious agent becomes more similar to the host cells, the more likely it is that there's going to be some toxic effects. So we have eukaryotic cells. If the microorganism we're dealing with is a eukaryote, it's going to be much more likely to cause us problems than if, say, we were dealing with a prokaryote. So complete selective toxicity becomes more difficult to achieve, and more side effects are seen, again, the closer that we are to the host cell. Now, some of the first attempts to develop antimicrobials, Robert Ehrlich wanted to develop a chemical compound that was a magic bullet, and you may have heard that term before, but the idea was that it would selectively kill bacterial pathogens. In his studies, he developed an arsenic compound called dioxydiamino arsenobenzo dihydrochloride, and thank goodness you don't have to learn that. It did have a couple of other names. It was arsphenamine, or phenamine in America, and it was called Salvarsan in German. It's also called compound 606. In any case, this was used in the treatment of syphilis. So there were 605 previous attempts before this one was used. On this particular substance, it was heated, not boiling, but certainly hot, and it was the male was catheterized, and this stuff was forced up the urethra and into the urinary tract. Gerhard Domack developed Prontosil. The active substance in this was later found to be the sulfonamides, sulfa drugs, and these were used in World War II and saved many lives. Alexander Fleming discovered penicillin in 1928. His research was actually kind of ignored for a little bit of time. In 1939, researchers found his report and were able to isolate the active ingredient, and thus penicillin became a very important antibiotic. In fact, many antibiotics still used today are derivatives. Certainly in dental practice, good old penicillin was used all the time. Now, generally speaking, the targets of antimicrobial drugs, there are several ways that these drugs can work. They can inhibit cell wall synthesis, and by inhibiting the cell wall synthesis, make the organism more vulnerable, more easily broken down. They can inhibit nucleic acid synthesis, which will interfere with cell division, and thus render the microorganism incapable of reproduction. Inhibition of protein synthesis. And since the function of microorganisms, since the function of all cells, really relies on proteins, both in structural and enzymatic actions, if you can inhibit protein synthesis, you can effectively destroy the cell. And then there is the disruption of cell membrane structure or function. So all of these are actually quite similar to the list of things that can occur that we saw with the physical means of control of microorganisms. If we take a look here, what you can see at the top, these are normal cocci, and they have this peptidoglycan layer. We have this exposed to a beta-lactam antibiotic, something like penicillin, which starts forming weak points in the peptidoglycan because there's no repair mechanism that can go on. If you then expose that to a hypotonic environment, that is an environment that has less solutes dissolved in it than are in the cell is going to absorb water, and ultimately the cell will expand and burst. So that's one activity for some antimicrobial drugs. Drugs that affect the bacterial cell wall. And keep in mind, most bacterial cell walls contain this girdle of peptidoglycan that we just looked at, and penicillin and cephalosporins block the synthesis of the peptidoglycan, causing the cell walls to lyse ultimately. Now, penicillins do not penetrate the outer membrane and are less effective against some gram-negative bacteria. There are some broad-spectrum penicillins and cephalosporins which can cross the cell wall of gram-negative bacteria. These broad spectrums have been modified, so we're looking at synthetics or semi-synthetic. There are drugs that inhibit nucleic acid synthesis, and they may block synthesis of nucleotides, inhibit replication, or stop transcription altogether. Now, sulfonamides and trimethoprim block enzymes required for DNA and RNA synthesis, so the cell's not capable of going through its binary fusion or mitosis or whatever process it uses. There is competitive inhibition that occurs here. And what happens is that the drug will compete with a normal substrate for an enzyme's active site, effectively blocking the enzyme from being able to bind what it's supposed to. There's also synergistic effects, an additive effect basically achieved by multiple drugs working together, requiring a lower dose of each, and this often happens where there is combination drug therapy. There are a number of drugs that block protein synthesis. There are a number of drugs that block protein synthesis. Ribosomes of eukaryotic cells differ in size and structure from prokaryotes, so antimicrobics usually have a selective action against prokaryotes. They can also damage the eukaryotic mitochondria, though. Some examples of these: the aminoglycosides, which include streptomycin and gentamicin, insert on sites of one of the subunits of the ribosomal RNA and cause a misreading of the messenger RNA. Tetracyclines block attachment of transfer RNA onto the acceptor site of the ribosomal RNA and stop any further synthesis. So, a couple of different activities. Now, I don't care if you know the exact method of activity here, but I would want you to know that tetracyclines and aminoglycosides are associated with blocking protein synthesis. There are drugs that disrupt cell membranes. There are drugs that disrupt cell membrane function. A cell with a damaged membrane dies from disruption in metabolism or lysis, just like we saw with the disruption of the cell wall. These drugs have a specificity for a particular microbial group based on differences in the type of lipids in their cell membranes. For example, polymyxins interact with phospholipids and cause leakage in gram-negative bacteria. Amphotericin B and nystatin form complexes with the sterols, steroid compounds, on fungal membranes, which cause leakage. As we can see in this graphic, some of the ways that this could occur. So we have polymyxin B, which makes it through the cell wall. It binds to certain sites on the cell membrane, ultimately disrupting the structure of the cell membrane and causing leakage. And at this point, we have completed the initial introduction to some of the characteristics of antimicrobials. Let's move on. We're going to stop here, and we're going to pick up on a survey of the major antimicrobial drug groups in a few minutes with our next presentation. So thank you.